Hi, everyone. I'm very happy to be presenting today some recent work from our lab that we are all very excited about. But before I actually get into this, I want to share with you the motivation for this project, as well as the motivation for our lab's research in general, and that's genetic disorders. So genetic disorders are rare diseases driven by mutations that drive a pathological change in the function of the affected gene. These are individually rare, but collectively they provide a large burden on society. There's over 7,000 genetic disorders, can affect up to 1 in 40 individuals. They're resulting for 20 to 30% of infant deaths and responsible for up to 50% of hospital-based pediatric practice. Also, there's limited therapeutic options here. Existing treatments often have a low therapeutic benefit, and due to the rarity of the individual disorders, the prices for these treatments are astronomically high, reaching up to $1 million a year for a treatment. And so one potential solution here is gene therapy. This is a therapeutic strategy where we're trying to treat a genetic disorder by directly addressing the underlying causative mutation. Traditional approaches function through the pathway of gene augmentation, where we're adding a functional copy of the defective gene. However, just as it's detrimental to have no functioning genes, it can also be detrimental to have too much. So dosing is a very important problem here. This makes gene editing, where we're directly and permanently correcting the causative mutation, a very appealing treatment strategy for the future. However, therapeutic gene editing has several challenges associated with it. The first of these is that we need molecular tools that can actually correct the causative mutation of the defective gene. And here, CRISPR-Cas9-based systems such as base editing and prime editing allow us to do this in a targetable, customizable, and universal framework. In the current state of this technology, we're able to efficiently perform all base pair conversions and small insertions and deletions. So this is letting us edit the genome in a way which even 10 years ago would have been considered science fiction. The second challenge is that we need delivery methods to actually bring gene editing tools to affected tissues. Two approaches commonly used are viral vectors and lipid nanoparticles. But when we look at what the ideal delivery vector would look like, we would want transient expression, non-immunogenic material, something with a low production cost, and something that can be targetable to affected tissues. And here, lipid nanoparticles plus RNA largely fit these criteria, really hitting on the first 3 points, but the fourth is currently a work in progress. And so these two fields have really exploded recently. If you look at PubMed publications, just searching for the terms lipid nanoparticles and CRISPR gene editing in 2022 alone, we had over 800 papers published for LNPs, over 2,400 for CRISPR gene editing. So there's really a rapid growth here. And what we need to evaluate all these emerging technologies is large screens. And unfortunately, we don't currently have a very effective model system for this. So our lab was interested in designing a model system for LNPs that deliver gene editing tools. When we think about what the ideal model system is, this is something that we can do in vivo, as in vitro results don't often predict in vivo performance. This is something that should tell us where gene editing is occurring. It should tell us how much gene editing is occurring, and it should have a fast, inexpensive, and detectable outlet, almost something that you can see by eye. And so what we took advantage of was the firefly luciferase gene, which is responsible for producing the bioluminescent signal that you would see in fireflies. We took a mutated copy of this that will produce a truncated and inactive luciferase protein and placed it in the mouse genome. When we correct this stop codon in the luciferase gene, this allows visualization of both the biodistribution and the efficacy of gene editing. One note about our model is we originally designed this for use with an adenine base editor, a tool which can convert AT base pairs to GC, making it applicable to about 50% of known pathogenic variants. And so we evaluated our model in an initial validation study. In this case, we're using adenine base editor mRNA delivered via an LNP, and we're going to assess editing in our mouse model over a period of 4 months using bioluminescence imaging. And then at terminal endpoint, we're going to take out the tissues analyze the luciferase activity, and also look at Sanger sequencing. Importantly, we decided to use 2 LNP formulations, which use currently clinically approved lipid nano ionizable lipids, which some of you may know as MC3 and ALC0315 from Onpattro and the recent BioNTech Pfizer vaccines, respectively. So our first experiment was actually looking at the quantification of these live imaging results. So what we found here was that following the injection on day 0, our luminescent signal remains stable for up to 4 months following a single injection of our LNP plus base editor RNA, indicating the potential of long-term gene correction with these tools. You can see from the quantification on the right that at each time point, the signal is fairly consistent. And we also see that it's localized within the region we suspect to be the liver. From this test alone, we were able to determine that the ALC0315 formulation was more effective than MC3, which is really what we're going for with this model, some sort of method to evaluate how different LNP formulations perform for gene editing. When we reached the terminal endpoint of 4 months, we wanted to assess the luciferase restoration within tissues. And here we used a luciferase assay where we're essentially taking out these tissues, homogenizing them, and looking at how much restored luciferase is present. We looked at tissues from the liver, spleen, lungs, and muscle. And what we saw here was that our ALC0315 treatment gave a restoration of around 84% in the liver. However, the MC3 treatment only showed about 8%. And due to the variability here, we actually didn't have statistical significance, although there is somewhat of a trend emerging. We also saw that there were no significant increases in luciferase restoration when we looked at the spleen, lungs, and muscle. However, this is largely to be expected as both of these ionizable lipids do promote liver hepatotropism. We followed this up with Sanger sequencing to get an idea of the actual editing in the liver at the DNA level. And so what we saw was that significant editing was only observed for the ALC0315 treatment. So you can see when you actually look at our Sanger sequencing outputs, at this target stop codon, the TGA, for our ALC0315 treatment, you're seeing the emergence of this corrected blue peak, which corresponds to the desired T to C base change. Looking at the actual quantification of this editing, we saw that the DNA correction for our ALC0315 treatment was around 25%, which is lower than our luciferase assay restoration. However, this could be due to a couple things. One is that protein restoration doesn't always match DNA correction. And 2, Sanger sequencing is also kind of a quick and dirty approach for the case of gene editing, as this is more of like a diagnostic technique rather than looking at actual percentages. So this is something we want to examine further in the future with next generation sequencing. And so for what we want to do with this model in the future, so far, I think what we have is that our model allows us to rapidly evaluate LNP formulations for in vivo gene editing. But what we can do with this is facilitate larger scale screens and inform more targeted LNP development. So what this might actually look like in an example is, let's say we want to target the muscle, central nervous system, skin, and maybe something that goes everywhere. You could start with an initial candidate LNP that delivers weakly to one of these areas. And then over successive rounds of optimization, we imagine that we could develop this lipid nanoparticle into something which shows strong efficacy for its initial target. Before I conclude, I want to address all the members of the Ross Lab that have contributed to this, as well as our funding sources. This really has been a team effort. I get to present the work, but so much work from other individuals has gone into this. And so just to give a final summary, recent developments in gene editing and delivery are really bringing us close to the treatment of genetic disorders, but we still have work to do. And so to help with this, we developed this luciferase reporter mouse model to facilitate rapid assessment of gene editing efficacy and biodistribution. And from our initial experiments, we were able to evaluate different LNP formulations for gene editing, which is what we want to do in the future. And with that, thank you everyone for listening.
Hi, Tyler. Would you be able to go back in your slides to that slide showing Onpattro BioNTech and just the, so what does that slide say about the relative efficacy of those two? Is the BioNTech gene editing is much more efficacious, or what was that actually saying? I didn't quite understand.
Was it this one?
Might have been the one before, it was either maybe the one after that one.
Or with
after
the
that one.
the sequencing?
Yeah, it might have been that one. Right. I think it might
Yeah.
have been that one. So what does that say about the three, the saline? Is that the control, or what have we talked, what happened there?
Yeah, so that was just mice which were injected with saline as our negative control. And so in
Right.
this case, this would be mice where we suspect that there's no editing occurring. So this is kind of what our baseline sequence looks like.
Right.
And then as we deliver the gene editing tools, we want to change this T to a C essentially.
Mhm.
And so kind of with the Sanger sequencing plot, you can see the emergence of this blue peak, which is present within the ALC 0315, but for MC3, like we're not really seeing anything above background. And so this is something like we were a little confused on, but we do want to probe it deeper in the future.
Right. So in a sense, the BioNTech formulation was the one that generated the transformation of a T gene to a C gene. Yeah.
Yes.
Protein. Yeah. Okay, great. Thanks for explaining that. That's really helpful.
Yeah, no problem.
Thanks. I hope you can hear me. Thanks for the presentation. It's really interesting work that you guys are doing. I have a question. When you checked for the DNA changes using the Sanger sequencing, did you take the whole liver, or did you take just a portion of it to check what percentage of the editing are you seeing?
Yeah, that is a really good point. So just in terms of actually generating the DNA sample and using our liver for other uses, we do only take a small portion of the liver for sequencing. We do try and get something that's similar across all the tissues we collect, but it could very well be that maybe we're sampling a portion of the liver where like certain cell types that were edited aren't particularly present. So that is actually something we were recently discussing, that we could go back to these liver samples and just try and get pieces from other areas to kind of see if this trend is holding up.
Okay, I guess I have a follow-up question. You did edited the luciferase, so you would be able to like see the whole the fluorescence from it. Did you observe difference in the fluorescence when you imaged the liver, like different portions of the liver, like lobes of the liver? Did you see a difference, or did you not check that, or
Yeah,
you didn't see any difference in the in terms of different lobes having different response to the gene editing?
yeah, so I don't actually have that data right here, but we do, like, as we take out the tissues at the terminal endpoint, we'll IVUS image the extracted organs. And what we do see is really it's a homogeneous signal throughout the liver. There doesn't seem to be any preference for any lobes.
Oh, okay.
Have you tested this model using any other routes of administration?
Yes, so didn't want to cram too much in here, but one of the things we're also interested in is delivery to the muscle. And so we did a very small pilot when we were first developing this model, where we tested this kind of approach with an intramuscular injection. And so here we used 5 different LNPs and just administered them into the gastrocnemius muscle of the mice. And what you can kind of see from this image is that we are able to determine that certain LNP formulations seem to be performing very well at editing in the muscle, whereas others we are seeing either faint or essentially no signal.
Could you provide a little bit more detail on how base editing works?
Yes, for sure. So in the next supplementary slide, we have the mechanism for base editing. And so with base editing, it's based on this CRISPR-Cas9 editing system, where you have essentially an RNA which will direct your CRISPR effector to a specific position in the genome. And in this case, instead of traditional CRISPR, where it's introducing double-stranded breaks, what they actually did was they knocked down this nuclease activity and attached deoxyadenosine deaminase onto the Cas9. And so this will bring this deaminase into contact with your target adenine that you're trying to drive the base pair conversion in. And so then the adenine deaminase is going to catalyze this reaction on the bottom, where adenosine is going to be, it's going to transition into inosine. And so you're going to have now an I in place at the A site. And then when DNA replication and repair occurs, the machinery is going to read our I as a G and then incorporate a C on the opposite strand. And this will eventually be edited out through successful generations.
Thank you so much.